okay, that can be used when you want to study about plant genetics or plant genomics. This will be very basic, okay, and I think I mentioned a few websites also during our lecture on Arabidopsis. Okay, for example, you can refer to certain database. Okay, for example, TAIR, right? TAIR. Okay, where you can get information or databases on related to Arabidopsis, for example. Okay, right. So just for introduction, okay. So database is actually web-based resources. We share information. Okay, so when scientists find some information, we akan deposit the data lah. Okay, right. So due to advances in genetics on plants, animals, okay, fruit flies, and other microorganisms. Okay, so scientists obtain a lot of information. Okay, so this information can be shared with other research community. Okay, and also with the advancement of computer programming. Okay, so we have this internet technology. Okay, so this contribute to the development of databases. Okay, so we talk about big data, bioinformatics. Okay, so based on this, we know that to do research, especially on genetic, we require these databases to store data. For example, we can store data on DNA sequences. Okay, so for example, you have information, you already sequence one organism. Okay, so you will share this information, these sequences with other research community. Okay, so you can also share database on mutant that you develop. Okay, so you boleh kongsi. Okay, you can also share literatures or paper yang you dah publish. Okay, with particular experiments. Okay, so one of the important database, okay, we call as NCBI. Okay, so which is actually National Center for Biological Information. Okay, so this NCBI contains several databases. Okay, yang you can use, okay, untuk check DNA sequences, RNA sequences, papers. So normally we use NCBI lah. Okay, so you can also search for scientific articles. Okay, using PubMed database, you can look for information about single gene. You can, and also in NCBI, it will link also to other resources, okay, within the NCBI website. Okay, so one popular page to visit at NCBI is what we call as the BLAST page. Okay, so BLAST stands for Basic Local Alignment Search Tool, which is actually you compare the genetic information using computer-driven mathematical formula. Okay, so for example, you sequence a gene. Okay, you sequence a gene, you dapat the sequence ATGC for example. Okay, so you want to check the mesh. Okay, sequence is similar dengan sequence gene apa for example. So using BLAST, you can input your sequence yang you want to know, and then you search to the close matches to the sequences in the database. Okay, so the sequences that are meshed are then written to the user in a list from most to the least similar. Okay, all right. Using BLAST also, you can convert this DNA sequence into amino acid sequence and also vice versa. Okay, so for example, from amino acid sequence, you can convert to DNA. All right. So this is example. Okay, you can go to the NCBI, lepas tu you click the BLAST page. Okay, so you boleh masukkan sequence yang you want to check, you want to cross match. Okay, so you letak the sequence here. Okay, and then you want to compare to database, you want to compare dengan human. Okay, you can compare also dengan mouse or you can compare with others. Okay, all right, so then you click BLAST. Okay, so bila you click BLAST, dia akan compare dengan database dalam available. Okay, so dia akan compare. Okay, for example, like I said, dia akan compare. This is for example, you don't know the sequence. Okay, you compare dengan dia akan compare dengan similarity. Okay, for example, the highest similarity, okay, dia akan tunjuk, okay, it's actually similar to Burkholderia, which is actually a bacterium. Okay, your sequence is actually similar to the sequence of Burkholderia. Okay, so this is what we say blast. Okay, and also dia akan keluar juga the similarity tu dia akan keluar dalam color. Okay, so you boleh compare. Okay, so the query is the one yang you masuk and the subject is yang ni lah, yang dia akan compare dengan Burkholderia. Okay, so dia akan compare the sequence similar atau tak. So this is what we call as blast. Okay. Alright, so ada website juga you boleh pergi which is Assemble. Okay, so this Assemble you boleh check sequence of human genome, you boleh check zebrafish, okay, you boleh search for all species lah. Okay, you can browse the genome. Okay, so ada banyak information yang boleh you dapat daripada website Assemble. Assemble pun ada function blast. Okay, so you boleh use this website lah. Okay, so the blast programs are actually terbagi kepada different type. Ada basically ada 5 type of blast programs. Okay, one is blast N, okay, which you submit. You want to query nucleotide sequence against a nucleotide database. Okay, N stand for nucleotide. Okay, blast P pula you submit to the database amino acid query sequence where you search against a protein database. Okay, so this is what we call as blast P. And blast X pula you submit nucleotide query sequence. Okay, you translate into all reading frames and you compare against protein database. Okay, so that is blast X. Okay, and then we have T blast N, which is protein coding sequence against a nucleotide database. Okay, translated in all reading frame. And then you have T blast X, which is 6-frame translation of a nucleotide coding sequence against the 6-frame translation of a nucleotide sequence database. So you boleh explore all these blast programs. Okay. Okay, any question? Okay, that is just some of the basic web based database yang you boleh guna lah. Okay, so I will give you 2 papers yang ada yang list different of database yang you boleh explore. Okay, you will learn more detail about this on subject of bioinformatics. Okay, about this web-based. Okay. All right, so I move to the next topic, which is genetically modified plant and also plant transformation. Okay, I think by now, okay, you should know, okay, what is genetically modified plant. Okay, so the definition, there are many definitions, okay, but this is the definition that I use. Okay, it's actually plant, okay, whose genome you already engineered in the lab, okay, in order to favor the expression of certain physiological trait. Okay, for example, when you transform the gene, okay, the gene confer resistance to cold weather. Okay, for example, or the trait that confer to the transform plant is actually drought resistance, for example. Okay, or you can transform a gene into the plant genome so that the plant will be able to produce certain compounds. Okay, biological products. For example, it can produce vaccine. Okay, it can produce proteins. Okay, it can produce enzymes. Okay, so this is the definition of genetically modified plant. Okay, any question? Tak ada? Okay. All right, so like I said, plant can be used as an expression system. Plant can be used as factory, okay, to produce many biological products, okay. But I would like to compare between other system, okay. So for example, you can use yeast also to produce biological products. Okay, you can use also bacteria to produce compound, okay, enzymes, vaccine. Okay, you can also use mammalian cell culture. Okay, you can also use transgenic animals. Okay, so but because we study plant, okay, of course plant offer other advantages lah compared to other system. Okay, so plant can be used as expression system, okay, to produce biological products. Okay, so this table to show you the comparison, okay, between other system, okay, as a factory to produce biological products. Okay, so when we compare in terms of cost, okay, if you use plant to produce a vaccine, for example, okay, the process is actually cheap, okay, compared to virus, also cheap. Okay, but if you use mammalian cell culture, okay, to produce biological products, mammalian cell culture is actually expensive. Okay, and transgenic animal also as a factory, okay, to produce biological product also expensive. Okay, and for storage, okay, if you develop transgenic plant that can produce vaccine, for example, okay, you can store the transgenic plant in room temperature. But for virus, it requires storage is -20. Yeast also -20, bacteria -20. For mammalian cell culture, you have to store in nitrogen, liquid nitrogen. Okay, so this is very expensive lah. Okay, and also for distribution. Okay, if you want to transport the transgenic plant, it's easy. Plant viruses also easy. Okay, but for mammalian cell culture, okay, for distribution is difficult and similar to transgenic animals. Okay, so the gene size, in terms of gene size yang you want to transform into plant, okay, not limited, but in plant viruses limited, yeast unknown, bacteria unknown, mammalian cell culture, the size of the gene to be transformed also limited. This is similar to animals. Okay, all right. And glycosylation, some protein to be active, okay, they need to be glycosylated. Okay, so glycosylation can happen in transgenic plant in the correct glycosylation process, which is similar to plant viruses, but yeast incorrect glycosylation can happen. So bila incorrect glycosylation, the protein that you express, okay, for example, vaccine yang you express will be wrongly glycosylated, so dia takkan function. Okay, bacteria, there is no process of glycosylation. So if you want to produce vaccine yang perlu process of glycosylation, so you cannot use bacteria as the system, okay, because bacteria cannot form glycosylation. Okay, mammalian cell culture is also correct, and also transgenic animal also correct. Okay, so these are some of the advantages of using transgenic plant lah, compared to other system untuk produce biological products. Okay, alright. So you boleh baca lah. Okay, alright. So this I want to show you. Okay, because we study plant, of course we want to promote plant lah as expression system. Okay lah, I said plant, if you use plant to produce biological product, it's economical because the process is cheap. Compared to microbial fermentation, the cost is lower, 2 to 10% of cost of microbial fermentation. And it's cheaper compared to mammalian cell culture because it only contributes 0.1% of the cost of mammalian cell culture system. Okay, and also if you want to express the protein, okay, you can express the protein at the targeted cells or tissue. Okay, for example, you want to increase production of vitamin A. So you boleh express the vitamin A only dekat seed atau kat grain because of the rice. Because apa yang kita makan adalah the grain, right? So you can express the protein at the targeted cell or tissue. Okay, and industrial scale production can be achieved if you use plant as expression system. Okay, because you can grow more of the transgenic plant. Okay, and also plant as expression system, there is less contamination of protein with human pathogenic microorganism. Okay, and also you can express this protein is the edible part. Macam kita kata tadi lah, you boleh express vitamin A dekat bahagian yang kita makan. Okay, or you can express protein dekat buah pisang. Okay, so that you boleh makan, kalau you want to express certain vitamin, you boleh dapat dari bahagian yang kita makan. Okay, and also another advantage of plant as expression system, okay, the process is easy and less expensive, and most importantly, plants can perform the correct post-translational modification, because like I said previously, some protein require to be active, it requires some modification towards the protein. Okay, so this can happen in plant because plant can perform most of post-translational modification. Okay, all right. Okay, any question? Sorry. Okay, any question?
Tak ada.
No.
Okay, all right. So now I want to teach you how to do plant transformation, how to do genetic modification. Okay, alright. So first, saya dah explain dalam tissue culture itu, but I will repeat. Okay, first you want to identify the gene that you want to transform into the plant. Okay, the interesting gene yang you want to transform into the plant. Okay, or what we call as the transgene, or what we call as gene of interest. Okay, so the gene of interest can be obtained from a donor organism. Okay, so for example, you want to introduce gene that can produce insulin. So you can isolate the gene from human. Okay, or you can introduce gene yang can be cold tolerance. So you can isolate from a bacteria that can survive on cold weather, for example. So the gene, interesting gene can be isolated from any donor organism. It can be from plant, it can be from bacteria, it can be from human, and so on. Okay. All right, so then you already know that, for example, you want to transform gene that can produce insulin into plant. Okay, so you isolate the interesting gene. Okay, then you clone into a vector or a plasmid. Okay, plasmid is actually naturally occurring DNA. Okay, extra chromosomal DNA can be found in bacterium. There's a plasmid, which we would use this plasmid. Okay, untuk genetic modification, you clone. Okay, you insert into a vector. Okay, and then you multiply the genetic construction. Normally dalam E. coli, you nak banyakkan lagi plasmid yang ada gene of interest tu. Okay, dalam E. coli. Okay, then you isolate the plasmid yang ada banyak with the gene of interest. You transform into, for example, agrobacterium. Okay, you introduce into agrobacterium, for example. Okay, because if you want to use agrobacterium technique lah, okay, to transform the gene. Okay, and then you transfer the gene. For example, in here dia pakai particle bombardment. Okay, so if you use agrobacterium, you infect the leaf explant with agrobacterium. Okay, and then you regenerate, okay, on a medium, okay, on containing selectable marker. For example, you add antibiotic. Okay, so those yang able to survive on that antibiotic, kita assume adalah yang transform. Okay, so then you evaluate the gene expression, okay, and then you do the crossing. So these are the steps for involved in genetic modification. Any questions so far?
Doctor, Yang lepas multiply guna E. coli tu kena masuk dalam agrobacterium dulu yang baru boleh letak dalam plant.
Okay, kalau you nak pakai method of agrobacterium.
Tak boleh ke E. coli terus letak dengan plant cell
Tak
tu?
boleh. Sebab E. coli dia tak infect, bukan dia tak infect plant, kita kena pakai special agrobacterium yang kita dah modify.
Oh, okay.
Okay, so kalau you nak pakai particle bombardment, after you multiply, you isolate the plasmid, you terus transform lah pakai particle bombardment. Tapi kalau you nak pakai, nanti saya akan tunjuklah method untuk transform plant. There are many method. One of the method is agrobacterium. Okay?
Okay, thank you, Doctor.
Any more question? Tak ada? Boleh follow? Okay? Alright. Alright. So, like I said, gene of interest yang tadi tu, you boleh obtain daripada bakteria, human, plants, of animal lah. Okay, so katakan in this diagram, they obtain the gene of interest from bacteria. So, they extract, isolate, and then clone into a vector lah. Okay. Alright, so after you dah clone semua, you can transform the gene of interest tadi either kepada nucleus or the chloroplast. Okay, alright, so there are the few advantages of chloroplast transformation which I will explain lah. So you boleh introduce your gene of interest into nucleus ataupun you boleh introduce your gene of interest into chloroplast. Okay. Alright, so how to introduce this gene of interest? Kita ada guna, you can use the technique of gene transfer via agrobacterium, or you can introduce the gene of interest via direct gene transfer, tak pakai agrobacterium. Okay, alright. Hopefully you can still follow me. Alright, so the first method is using agrobacterium mediated. Okay, macam kita transform the first experiment hari tu. Okay, kita dah multiply the plasmid, okay, with the gene of interest dalam E. coli. Okay, then kita isolate the plasmid, kita transform into agrobacterium. Okay, so the concept of agrobacterium, okay, in nature, there are two types of agrobacterium. Okay, one is we call as agrobacterium tumefaciens. Okay, and the other one, agrobacterium rhizogenes. Okay, so you have to understand the biology of agrobacterium first. Okay, so agrobacterium tumefaciens ada plasmid what we call as TI plasmid. TI stand for tumor-inducing plasmid. So in nature, kalau this agrobacterium infect the plant, okay, dia akan cause formation of tumor. Okay. And another type of agrobacterium is agrobacterium rhizogenes. This agrobacterium contain Ri plasmid, which is root-inducing plasmid. So in nature, if agrobacterium rhizogenes ni, kalau infect the plant, dia akan menyebabkan plant ni produce more roots. Okay? Alright, so agrobacterium tumefaciens, okay, kita use agrobacterium tumefaciens for transformation. Is actually soil-borne. Dia akan dijumpai kat soil. It's actually gram-negative bacterium. Like I said just now, if plant is being infected with agrobacterium tumefaciens, it will cause tumor or what we call as crown gall disease. Okay, so this is the tumor kalau di infect. Okay, so the crown gall formation depends on the presence of the TI plasmid. Okay, any question? So based on this, scientists modified the agrobacterium tumefaciens. Alright. So at the TI plasmid, okay, in nature, ni dah diubah, okay, scientists dah ubah, the TI plasmid sekarang ada tDNA regions, okay, tDNA region here. Okay, so when you do the transformation, anything between the tDNA region will be transferred into the plant genome. Okay, so your gene of interest should be are located at the tDNA region. Okay, any question? Okay, in nature, TI plasmid also contain virulent region. Virulence regions ni adalah genes yang perlu untuk menyebabkan jangkitan. Okay, and TI plasmid also contain origin of replication for the replication of the plasmid. Okay, and also contain a region enabling conjugative transfer. Okay, alright, conjugal plasmid transfer, and also contain genes for the catabolism of opines. Okay, so this is in TI plasmid, what you can find in TI plasmid. Okay, so based on this concept, scientists modified this TI plasmid to be used for genetic transformation of plant. Okay, clear? Any question? Any question? Tak ada? Boleh follow Okay, so for transformation using agrobacterium, okay, there are two types of vector. Okay, so kita guna one type of vector we call as co-integrative vector, and another type of vector yang sekarang kita guna ialah binary vector. Okay, so the difference between co-integrative vector and binary vector, as you can see, in co-integrative vector, the tDNA region, okay, here is located on the same plasmid with virulent regions. This is co-integrative vector. Okay, so nowadays we hardly use co-integrative vector. Okay, kita sekarang guna binary vectors. Okay, where we have two plasmid. One plasmid contain the tDNA region with gene of interest. Okay, while another plasmid contain the virulent regions. Okay, so nowadays we use this binary vector. Okay, where the virulent region is located on different plasmid from tDNA region. Okay, any question?
No.
Tak ada? Okay, boleh follow Hopefully tak berat sangat lah Okay, so this is example yang hari tu kita guna untuk kita punya transformation P-Cambia vector. As you can see, P-Cambia vector is actually binary vector because you cannot see the virulent region. Virulent region is located on another plasmid. Okay, like I said, any gene of interest should be located between the tDNA region. So this tDNA region is being flanked with T border left and T border right. Okay, so it contains multiple cloning site where you can clone your gene of interest or you can insert your gene interest, and then there are the selection gene lah. It can be antibiotic resistant, it can be herbicide resistant, for example. Okay, to select transform plant. Okay, I think you are quite familiar lah because you done the experiment one tu kan. Okay, any question?
Doktor, yang apa tu binary vector kan yang ada satu TI plasmid, satu lagi binary vector. Yang co vector tu co-integrative vector tu kayaknya boleh je lah kalau sumber dua benda tu dari sumber yang berlainan, lepas tu masukkan dalam agrobacterium. Yang binary vector dengan TI plasmid tu memang ada dalam agrobacterium tu ke boleh
agrobacterium. You can see this is agrobacterium cell. Tapi dia dua plasmid. Satu carry the tDNA region, satu carry virulence region. This is berbeza dengan co-integrative vector, which is the virulent region dengan tDNA region located on the same plasmid. Boleh? Okay, nampak?
Faham.
Sebab virulence region ni diperlukan untuk menjangkiti pokok tu. So bila you incubate plant yang you want to transform, okay, virulence region ni the function is untuk menyebabkan jangkitan. Virulence, okay, jangkitan. Alright, so dia perlu ada. Tapi for co-integrative vector, the virulence region dengan tDNA located on the same plasmid. Okay. But for binary vector, virulent region dalam TI plasmid, but tDNA dalam binary vector, satu plasmid lain. Okay, boleh ke?
Lepas tu kan agrobacterium tu sebagai dia punya apa macam medium kan. Lepas tu apa plasmid tu kan kita nak masukkan dalam agrobacterium kan. Kan. So plasmid tu yang plasmid yang binary vector dengan plasmid TI plasmid tu boleh je lah dari sumber yang berlainan. Macam tu boleh ke?
Sumber berlainan.
Contohlah kononnya lah jin untuk yang plant resistant tu kita dapat daripada satu bakteria ni nama dia A. Lepas tu tapi dia ada yang apa tapi dia tak ada virulence region tu. Lepas tu yang
ini contain selectable marker dengan gene of interest. tDNA ini selectable marker dengan gene of interest. Okay. So virulence ni tak perlu ada selectable marker lagi dah. Maksud apa? Your gene of interest, you boleh dapat daripada human, gene apa yang you berminat lah nak introduce dalam plant, betul tak? Virulence region ni adalah virulence region of course daripada agrobacterium lah, sebab agrobacterium yang nak menjangkiti plant tu.
Oh ya ke? Okay, faham.
Kan? Okay, what you need to multiply is sekarang ni adalah inilah yang gene of interest tu lah, yang ada gene of interest tu lah. You multiply dalam E. coli dulu, lepas tu you isolate, masukkan dalam agrobacterium.
Okay, faham.
Yang you want to transform, boleh?
Boleh.
Boleh? Clear?
Clear.
Okay. Alright, so this is binary vector, okay, because this is for Picambia lah. Okay, so you can see that to drive the expression of gene of interest, of course you need promoter. Okay, so that the gene will be expressed lah. Okay, so the gene will express to produce insulin, for example. Okay, so there are few types of promoter. Okay, so the different type of promoter, you know what is a promoter, right? Apa dia promoter? Apa promoter? Okay, what is a promoter?
Untuk nak bindkan nak dibind dekat DNA region tu, lepas tu baru boleh masuk kan.
What is a promoter? Okay, this is very basic tau. That's why saya kata genetics ni you kena ada asas plant biochem semua tu. What is a promoter? When we talk what is a promoter in terms of DNA, what is a promoter? Okay, siapa boleh jawab? What is a promoter?
Ya, untuk introduce gene. Tak tahu.
What is a promoter? It's a basic you belajar ni, form 5 semua ni.
Untuk bind DNA.
Apa dia? A promoter is a site where is a site where transcription starts. Dah lupa, it's a basic, you know. It's a promoter, is a site, a sequence that will be recognized for transcription to start. Okay? Ingat ke? Boleh? Still remember what is a promoter? Okay, alright, boleh Okay, jangan lupa. Okay, so dia akan get more complicated. So, bila you dah identify gene insulin gene kan? Okay, for example, then you need to clone the promoter. What type of promoter do you want to use? Okay, so untuk plant transformation. Okay, so one common promoter yang being used to drive the expression of your gene of interest is we call as agrobacterium derived promoter. Sorry, please be quiet. What do you want? Computer, please share. Okay, is actually one type of promoter. There are different type of promoter that can be used. Okay, listen carefully. I will explain the different promoter yang you boleh use to drive the expression of your gene of interest. Okay, one of them is actually promoter yang you isolate from agrobacterium. Okay, so you can use agrobacterium. Okay, dalam agrobacterium dia ada NOS gene. Okay, dalam agrobacterium ada NOS gene. So you boleh ambil the sequence of NOS gene punya promoter and then you clone sebelum you punya gene of interest. Okay, so this NOS gene punya promoter can be used to drive expression of your gene of interest. Okay, so this type of promoter is considered constitutive, which is your gene of interest tadi tu bila masuk dalam plant, okay, dia akan diekspres setiap masa. Okay, dan akan diekspres throughout the plant. Okay, so this is what we call as constitutive promoter. Okay, boleh follow lagi tak? Any question? Boleh? Okay, kalau tak faham, I will explain slowly. Okay, the second type of promoter, what we call as constitutive promoter. Okay, so this promoter akan drive high expression level in all cell types throughout the entire growth and development period. Okay, so kalau you nak, bila you transform dalam plant, gene A ni, for example, insulin gene, you nak insulin gene diproduce dalam plant dengan banyak. Okay, so we use constitutive promoter because this type of promoter akan overproduce protein of interest. Okay, if you want to study function of gene also, you can use a constitutive promoter. Okay, so one example of constitutive promoter is a promoter that you obtain from virus 35S RNA gene, cauliflower mosaic virus 35S RNA gene. So dalam cauliflower mosaic virus, dia ada 35S RNA gene. So scientists isolate the promoter, the sequence of promoter of RNA 35S RNA gene ni, and then clone sebelum gene of interest dalam plant, dalam vector plant. Okay, so that bila gene of interest ni masuk dalam plant, okay, the gene ni akan di-express constitutively setiap masa, okay, throughout the growth, throughout the all cell types. Okay, alright, any question? Tak ada? Okay, other example of constitutive promoter where you can obtain from actin genes of rice. You can also obtain this promoter from brachypodium elongation factor gene and ubiquitin gene. And also you can obtain this promoter from switchgrass ubiquitin gene. Okay, so this gene produce promoter, what we call as constitutive promoter. Okay, any question? Boleh faham kan? Tak ada soalan? Boleh? Boleh follow Okay, then we have, boleh faham? Okay, then we have tissue specific promoter. This is where you use this type of promoter where you want certain part of the plant to express your gene of interest. You tak nak semua part of the plant express your gene of interest. Okay, so this type of tissue specific promoter is being used to limit the expression of any potentially harmful substances to tissues that are not consumed by humans and animals. Or you want to also, this type of promoter can be used so that only certain tissue will express your gene of interest. Okay, so macam saya kata tadi lah, kalau you want to produce vitamin A dekat endosperm, dekat grain, so you can use endosperm specific promoter. So your vitamin A ni, the gene will be produced only dekat endosperm saja. Okay, alright, or we will also produce dekat seed saja. That's why we use tissue specific promoter. Okay. And the final type of promoter is what we call as inducible promoter, where the expression of the gene, okay, the gene will be expressed, your gene of being expressed only when it's being induced because you use inducible promoter. Okay, so this promoter will be active only, for example, it being induced. Okay, so for example, it can be induced by chemical factor. For example, for example, you already transform this planet with gene of interest yang you include promoter will be induced with tetracycline. Okay, so you can spray the transform plant dengan tetracycline, for example, you can grow on tetracycline, and this promoter will be active and your gene of interest will be transcribed and translated. Okay, so this is what we call as inducible promoter. Okay, this promoter akan be active. One is being induced by chemical factor, or it can be induced when exposed to certain condition, for example, exposed to heat, exposed to cold or light. Okay, so this is what we call as inducible promoter. Kita tak nak gene of interest dia express sentiasa, which is, this is the difference between constitutive promoter and inducible promoter. Okay, any question? Tak ada, faham? So, inducible promoter can actually be divided into 3 categories. Okay, the first one is non-plant derived system, where you want to induce this promoter is by exposing the transformed plant with a certain chemical. For example, you expose the plant with alcohol. Okay, you can induce the promoter, okay, to be active by exposing to copper. Okay, so this is what we call as non-plant derived system. Okay, so another category of system of inducible promoter is what we call as a response to environmental signal. Okay, for example, you induce the promoter when it's being exposed to wound. So you need to lukakan sikit the transform plant so that the promoter will be active. Okay, or you can expose the plant to heat shock, to heat. Okay, so that the promoter will be active. Okay, and the final category of inducible promoter is called plant derived system, where the promoter is being active when you is being depending on the developmental stage of the plant. Okay, so for example, in this case, the promoter will be active when the plant, the transform plant, dia arrive at the senescence stage. Okay, so the promoter will be active. Okay, so these are the example of inducible promoter. Okay, so that is agrobacterium. Okay, all right. Can I stop for 5 minutes? Okay, for break. Then I continue with another method how to transform gene of interest using direct gene transfer using particle bombardment. Boleh?
Boleh.
Okay, kita stop dulu. kita start dalam pukul 11. Boleh?
Kira
Boleh.
time.
Okay.